and welcome to Spotlight on Antibodies, a podcast from BioIntron where we bring you the latest antibody industry trends. This month in February, we'll highlight radioantibodies, antibody cytokine fusion proteins, and conditionally active antibodies. Let's start with something that's been gaining some momentum in oncology over the past few years: radioligand therapy, often shortened to RLT. At its core, radioligand therapy is a way of delivering radiation directly to cancer cells, instead of bathing large areas of the body in radiation, as in traditional radiotherapy. So it works as radioligands are made of two main parts. First, there's a targeting molecule. This can be a small molecule, a peptide, or increasingly, an antibody. Its job is to recognize and bind to a specific marker on cancer cells, like a molecular address label. Second, attached to that targeting molecule is a radioactive isotope, which delivers the therapeutic radiation. When the targeting molecule finds its marker on a tumor cell, it brings the radioactive payload with it, allowing radiation to be delivered exactly where it's needed, while minimizing damage to surrounding healthy tissue. The very first injected radiotherapy dates back nearly a century, when radioiodine was used to treat thyroid disease in the 1930s and 1940s. What's changed since then isn't the concept, it's the precision. Today, instead of targeting entire organs, researchers can hone in on specific molecular markers expressed by cancer cells. That's where antibodies enter the picture, as antibodies are naturally precise targeting tools. In radioligand therapy, radioantibodies use the specificity to guide radiation straight to tumors. This approach has been especially powerful in cancers like prostate cancer and neuroendocrine tumors, where well-defined surface markers are available. One of the most exciting areas of progress right now is the radioactive isotopes themselves. Many RLTs have used beta-emitting isotopes, which release particles that travel a few millimeters through tissue. That can be useful for larger or heterogeneous tumors, but it also increases the risk of collateral damage. More recently, there's growing interest in alpha emitters, radioisotopes that release very high-energy particles over extremely short distances, often just a few cell diameters. In simple terms, alpha particles are like a sledgehammer with a very short reach, devastating to the targeted cell, but far less likely to affect neighboring healthy tissue. An example of this trend is recent work in PSMA-targeted prostate cancer therapies. For example, a program used the alpha emitter actinium-225. Clinical experience across more than 100 patients had shown that encouraging anti-tumor activity helped to limit common side effects seen in earlier PSMA-directed radiotherapies, such as salivary gland and kidney uptake. Other than alpha emitters, researchers are also exploring beta emitters, such as terbium-161. What makes terbium-161 particularly interesting is that it combines traditional beta radiation with Auger electrons, which deposit energy over extremely short distances. This creates a hybrid effect, maintaining good imagery and dosimetry, while potentially increasing tumor cell killing at the microscopic level. How the payload is attached to the antibody also matters enormously. That's where advances in conjugation chemistry come in. Newer platforms can allow scientists to attach radioisotopes at defined sites on the antibody, typically in the Fc region, which is the part of the antibody that doesn't bind to the tumor. This avoids interfering with target recognition and produces more predictable drug products. This matters because in radioligand therapy, small differences in how an isotope is attached can influence how long the antibody circulates, how well it binds the tumor, and how radiation dose is distributed in the body. We're now seeing radio-labeled antibodies and antibody-derived formats, like nanobodies, VHH antibodies, bispecific antibodies, and engineered fragments, being tested across a wide range of targets, including PSMA, HER2, EGFR, and c-Met. Some are designed primarily for therapy, others for imaging, and many are built as theranostics, meaning the same targeting molecule can be used first to image a tumor and then to treat it. Looking ahead, radioligand therapy is evolving into a platform modality, one that combines the force of radiation with the molecular precision of antibody engineering. In this next section, we're shifting focus from radiation to immune signaling. Specifically, we're looking at antibody-cytokine fusion proteins, a class of molecules designed to harness the power of cytokines while controlling where and how that power is applied. Cytokines are short-lived signaling proteins that immune cells use to coordinate activation, proliferation, migration, and function. In normal physiology, cytokine signaling is often local and tightly regulated. However, when cytokines such as IL-2, IL-12, IL-15, or IL-21 are administered systemically for therapy, that natural spatial control can be lost. Because cytokine receptors, or key receptor subunits, are broadly expressed across immune cell populations, systemic dosing can lead to widespread receptor engagement and dose-limiting inflammatory toxicities. By physically linking a cytokine to an antibody that binds a defined cell type, tissue feature, or disease-associated marker, these constructs aim to improve the therapeutic index through spatial restriction of signaling. Recent work in this area converges around a few main engineering strategies. One approach, often referred to as cis-targeting, uses the antibody domain to bind a specific immune cell subset and deliver cytokine signaling, preferentially to that same cell. That is particularly relevant for common gamma-chain cytokines, which share receptor components and activate JAK-STAT signaling pathways. Because these receptors are widely distributed, systemic cytokine administration activates both intended and unintended cell populations. Cis-targeted immunocytokines increase functional selectivity by coupling antibody binding and cytokine signaling in close proximity, reducing activation of bystander cells. A second theme is that molecular architecture matters. Studies show that cytokine positioning and antibody format can substantially influence signaling output. In tumor-targeted constructs combining IL-15 with IL-7 or IL-21, cooperative cytokine effects were strongest when the fusion proteins were bound to their target. Different cytokine pairings also produced distinct immune outcomes. Some constructs favored interferon gamma production and cytotoxic activity, while others preferentially supported naive T-cell proliferation. A third strategy uses disease-associated markers as anchors to retain cytokines locally. In one example, CAR T cells were engineered to secrete an anti-PD-L1 IL-12 fusion protein. Because PD-L1 is inducibly expressed in the tumor microenvironment, the fusion protein preferentially localizes IL-12 to tumor tissue. Compared with non-targeted IL-12 secretion, PD-L1 anchored IL-12 increased intratumoral immune activation and T-cell infiltration while reducing systemic inflammatory effects. This work illustrates how checkpoint ligands can function as spatial docking sites for cytokine delivery. Similar localization strategies are now being explored beyond oncology, including in infectious disease models, where targeted cytokine or chemokine delivery can restore deficient immune responses at sites of infection. Across these examples, antibodies primarily serve as spatial regulators of cytokine activity. By controlling where cytokines engage the receptors, antibody-cytokine fusion proteins aim to preserve immunostimulatory benefit while reducing systemic toxicity. Many antibody drug targets present a fundamental challenge. They are biologically important but broadly expressed. Targets like immune checkpoints, co-stimulatory receptors, and certain tumor-associated antigens cannot always be engaged at therapeutically optimal doses without triggering on-target, off-target toxicities. As a result, systemic exposure is often limited not by lack of efficacy but by safety. One response to this problem is the development of conditionally active antibodies. which are antibodies engineered to remain functionally restrained under normal systemic conditions and become fully active only in specific biological contexts, such as the tumor microenvironment. A conditionally active antibody is engineered so that one or more of its key functions, like antigen binding, receptor clustering, or immune effector recruitment, is reduced at baseline and restored only after exposure to a defined trigger. There are two common ways that this is achieved. One is using masking strategies. Here, part of the antibody's binding surface is physically blocked by a removable domain. This mask is typically released by tumor-associated proteases, which are enzymes that are more active in diseased tissue than in healthy tissue. The second approach relies on spatial or coincidence logic, where productive signaling requires simultaneous engagement of multiple targets or anchoring to a specific cell surface. In these designs, the antibody is only functionally active when it is positioned in the right microenvironment. Checkpoint inhibitors are an example of why conditional activation is attractive. Antibodies against CTLA-4 can be effective, but are associated with immune-related adverse events when administered systemically. In a recent preclinical study, researchers engineered a conditionally active anti-CTLA-4 antibody using a protease cleavable design, intended to restrict activation to the tumor microenvironment. The antibody remained functionally attenuated until tumor-associated proteases removed the inhibitory domain. In mouse models, the fully cleavable version slowed tumor growth comparably to standard anti-CTLA-4 therapy, but was associated with less treatment-related weight loss, a common proxy for systemic toxicity in preclinical studies. A partially cleavable version, which remained tethered to its targeting element after activation, showed reduced efficacy. Increasing tumor localization does not automatically guarantee optimal functional activity once inside the tumor, where diffusion, receptor engagement, and local concentration all matter. Importantly, these results support the idea that conditional checkpoint blockade is primarily a safety-driven strategy aimed at preserving efficacy while reducing systemic immune activation. Masking strategies themselves have evolved, as earlier approaches often relied on non-human masking domains, raising concerns about immunogenicity. More recent designs use human-derived components. One example uses a calmodulin-based peptide clamp system, in which masking domains are attached to the antibody through protease-cleavable linkers. In masked form, antibodies such as trastuzumab and cetuximab showed markedly reduced antigen binding and diminished antibody-dependent cellular cytotoxicity. After exposure to tumor-associated proteases, both binding and effector function were restored to near-native levels. Conditionality does not always require protease activation. In some bispecific antibodies, activity can be controlled through anchoring-dependent receptor clustering. In one c-Met and CD137 bispecific design, CD137 agonism was engineered to occur only when the antibody was anchored to tumor cells via c-Met binding. Receptor clustering was minimal unless both targets were present. This approach localizes co-stimulatory signaling to the tumor and microenvironment and addresses toxicity concerns that have historically limited systemic CD137 agonists. The concept of conditional activity has also been applied to antibody-drug conjugates, or ADCs. In a first-in-human study of a ROR2-directed ADC described as a conditionally active, high response rates were reported in a small cohort of patients with advanced melanoma. While dose adjustments were required for known toxicities, treatment discontinuation due to adverse reactions were not reported in this early study. Finally, computational protein design is beginning to influence the space. Recent studies have shown that de novo designed peptide masks can be engineered to sterically block binding interfaces and be removed by protease cleavage. Even relatively low affinity masks were sufficient to suppress activity when tethered, with binding restored after cleavage. This work highlights how proximity effects and rational design can accelerate the development of modular conditional systems. And that's all for this month's Antibody Industry Trends. To learn more insights into antibody drug news, make sure to follow BioWintron or LinkedIn on our website at www.biowintron.com. Keep up to date with further episodes from Spotlight on Antibodies by following this podcast.